The recording begins. It is November 3rd, 2025. This is Biology 105. Thank you all for being here today. Today we are going to talk about meiosis. Then next week we talk about genetics, mainly from the perspective of inheritance. So we're going to talk about inheritance patterns, how to predict phenotypes, Punnett squares. I'm going to tell you that in this class, I don't really like Punnett squares. I'm going to show you how to never have to use one again. Thank you. A Punnett square is just a way to predict the likelihood of multiple independent events occurring at once. And we have math for doing that. You don't need the Punnett square because you lean against it, you lean on it too much, it gets bigger and bigger and unwieldy, and then you tip over the other way. All right? So we're going to talk about probability rules here, and it's going to be so much fun. Anyway, thank you. I'm so happy that you're excited. You may not know it, but all of you are excited about not having to use Punnett squares. A statement doesn't make a ton of sense, but don't worry about it. Just move on. Just move on, John. Week 11. It's already forgotten. Week 11, here we are. Meiosis, we've been talking about mitosis. Mitoses go in my sockses, very funny. Mitosis, now let's talk about meiosis, where we generate gametes for sexual reproduction. The process of meiosis and then fertilization is going to lead into our discussion of what you inherit from each parent, the likelihood of inheriting a characteristic, terminology like homozygous dominant, homozygous recessive, heterozygous, things that you've probably heard about. But then make a note here that in week 13, the week before your fall break on Monday, you do have exam 3. And this exam is going to be basically exclusively on mitosis, meiosis, and patterns of inheritance because they're so important. Yes?
You said basically, does that mean?
Yeah, totally. Let's look at the study guide.
I can also just look at the study guide.
You can always just look at the study guide. I love my little study guides. All right, here we go. Quantity of DNA at each stage of interphase, where that wide nuclear envelope degrades, asexual reproduction, homologous chromosomes, Punnett squares. Interpret these terms. Yep, it's all about mitosis, meiosis, and patterns of inheritance. So the basically can be lost. Okay, any questions about that? We got the standard stuff of like, keep going to your labs, keep completing your reading quizzes. Any questions, comments, concerns? Yeah, please.
So I'm assuming this next exam is going to go really in depth on those topics?
Exactly. So check the study guide. I tried to make the study guide reflect that. Same length, but yeah, only 3 subjects or 3 PowerPoints really to be tested on. All right, yeah, no problem. Any other questions? Okay, well, I'm very excited to just go ahead and get started. We'll wrap up the last of our mitosis slides right here. Bless you. And then jump into our meiosis slides. So I finished last Friday by talking to you about differences in cytokinesis between animal cells and plant cells. The main difference being that you've got this dang cell wall that needs to form between plant cells before they finish dividing. All right? Okay, Taxol. Taxol is a potent anti-cancer drug. It's extracted from the Pacific yew tree. We've got Pacific yews all over the dang place here, but they're relatively rare and the concentration of Taxol within them is relatively low. What does Taxol do? Well, Taxol inhibits microtubule formation. So if Taxol disrupts microtubule formation by actually making them overextend. They get unwieldy. All right. So you have the 2 microtubules are too long. They kind of fall apart. They can't do what they're supposed to do. So what would this, if you're not allowed to make microtubules, okay, then what would this affect? A, the formation of the mitotic spindle. Remember, That's when the microtubule attaches to the centromere of the chromosomes, all right? And then, and that allows the chromosomes to get moved about during M phase. If there's no kinetochores formed, then the chromosomes can't align cannot align at the metaphase plate, and the sister chromatids can't be separated if the mitotic spindles aren't present. Therefore, the cells don't divide. What's one of the key characteristics of cancerous cells? They divide uncontrollably. But if you can't form mitotic spindles, the cells can't divide. All right? So there you go. The eukaryotic cell cycle is regulated by a molecular control system. So you ever use one of those mechanical light timers where it's a little wheel that spins and you lift up or you put in these little metal tabs and there's actually a little switch that goes click, click, and you hear it go, if you listen really closely, you know what I'm talking about? Yeah, some people do, some people don't, and you've just exposed yourselves if you know what that is. No, it's a joke, it's a fun joke. Okay, so check it out. Basically, this is modeled after one of those. Okay, there's this little bar that's trying to spin around for the cell cycle to proceed, and there's these little barriers popped up here. Okay, these barriers get dropped if the cell meets certain requirements, and then the cell can proceed through the cell cycle. So this is going to be really basic overview of this stuff. But the point is that the cell is making sure that it has enough resources, that the chromosomes are being replicated correctly, that all the mitotic spindles have formed, all right? There's a bunch of different intracellular and extracellular signals, bless you, that the cell detects, and that allows the cell to proceed through its cell cycle. So these extracellular signals include things like growth factors, growth hormones, which will be like, oh yeah, push the cell through, like go, go, go. You need to be dividing right now. We want to grow as much as possible. But I'm going to talk about one specific type of intracellular receptor, which is the cyclin-dependent kinase and cyclin system. And you already know, cyclin-dependent kinase, that's an enzyme that transfers phosphate groups from ATP onto other things. It probably makes stuff happen, and it's dependent on cyclin. Whatever cyclin is, we'll talk about that later. Okay, different cells divide at different rates. They proceed through the cell cycle at different rates. These checkpoints are points where the cell is checked for adequate resources and that the essential processes have completed for the cell to proceed. All right. So you have a checkpoint in the G1 phase. This G1 checkpoint, make sure the cell's big enough, make sure the cell has enough resources, all right? Checks for genomic DNA damage. So is there damage to the genome? Are the sequences off on anything? How do these things get checked? It's usually through like mispair detection. If you have, when there should be an A and a T, instead you have an A and a C, that can be detected. There's like a weird shape change in the molecule. But how any of this operates is well beyond our concerns as a Biology 105 class. You'll talk about it in a genetics course or a cell biology course. We're just going to go real basic on these checkpoints. I hope that's okay for everybody. So you're going to see some commonalities here where a lot of these checkpoints involve making sure there's enough cell material present. And then if DNA can't be repaired, because there are mechanisms inside of cells to repair DNA, if DNA can't be repaired, the cell will usually undergo apoptosis, which is controlled cell death, right? Solid stuff. There's this G0 phase, and this G0 phase is basically like, just get out of here. We don't want you to keep dividing. You're done. All right. The cell might enter G0 and then go into apoptosis, or maybe the cell could possibly leave G0 and resume G1, but it's not something to be too concerned with currently. All right. So you got this G1 checkpoint. Does the cell have enough stuff inside of it? Does the DNA seem to be okay? All right, do we have any major detectable mismatches or damage to the DNA? Then we've got a G2 checkpoint. So what's happened at this point? Well, the cell's finished G1 and new DNA has been synthesized during S phase. So now all the chromosomes are duplicated or replicated. All right, so the G2 checkpoint is going to make sure that the chromosomes have all been replicated. It's also going to make sure that the cell has adequate resources and that the cell is of an adequate size. How are these things detected? It's a lot of stuff. All right, we're not going to worry about any intracellular signals beyond cyclin and the cyclin-dependent kinases. But that will give you an idea of how this system might operate. Then there's an additional checkpoint during the M phase. And this checkpoint makes sure that all the chromosomes are attached to the mitotic spindle. So the cell won't proceed through mitosis or meiosis unless all of the kinetochores have formed. All right? If the kinetochores haven't all formed, that means that the chromosomes aren't going to be separated or aren't going to be moved, separated, allocated correctly, and we'll end up with daughter cells that have abnormal numbers of chromosomes, and that usually leads to a non-viable cell. All right. Awesome. Questions about these checkpoints? There's really not. We're really not going into much detail. I just want you to know that the cell's checking. All right. And now let's talk about one, because usually the question is how does this operate on the intracellular level? And we'll talk about one system, one specific system that's involved in regulating the cell cycle internally. And this is the cyclin-dependent kinases and cyclins and maturation promotion factor. Okay? So check this out. You've got this enzyme called a cyclin-dependent kinase, and it's actually a family of enzymes. All right? They're kinases, so you know that they phosphorylate things. And usually when you phosphorylate things, you are providing energy to make stuff happen. In this case, you're providing energy to make the cell division happen. All right? Now, these cyclin-dependent kinases are present at a relatively high level in the cell all the time. The level of cyclin in a cell is a flat line, okay? There's always The level of the cyclin-dependent kinase. I really got to slow myself down and use the correct language here. The level of the cyclin-dependent kinase, the enzyme, is constant throughout the cell cycle, okay? What changes is the concentration of cyclin, all right? So cyclin accumulates sort of beginning at the end of the S phase, all right? Here we go, cyclin concentration right here. Beginning at the end of the S phase, it starts to accumulate through the G2 phase, and then in the M phase, it reaches its highest level, and then it starts to be degraded so that there's low to none of it inside in the cells that result from the cell division. So this means that even though the concentration of cyclin-dependent kinase is constant throughout the cell cycle, the cyclin-dependent kinase's activity goes up beginning in G1 and hits its peak in the M phase, all right? So this is how the cell regulates cyclin-dependent kinase activity, and an activated cyclin-dependent kinase it transfers phosphate groups in ways that encourage the processes of the M phase. It encourages you to move through the G2 and move through the M checkpoints, all right? And you might wonder, like, well, why does any of this happen this way? Well, think about this, all right? The example that I'll usually use is you are making a complicated, assembly of gears. There's 10 gears you have to put together, right? And you jam them all together, And then when you're done, and then you're done, somebody needs to make sure that you have enough complicated assemblies of gears. Let's say you're doing 12 different assemblies, and you need to make sure that there are 50 of each of those. You could count all of those, or every time I finish making something, I could throw a ping pong ball into a bucket, all right? So the ping pong ball goes into the bucket, and then once that ping pong ball gets heavy enough and it weighs down the little thing, it goes ding, and then I get to go home for the day. And we go and we divide, all right? So the cyclin can be sort of a byproduct of other essential reactions, where then they just make cyclin. It's like, I'm making this complicated thing, but I'm also going to be making cyclin to give me a rough idea of how much stuff I've done, how many other things I've accrued. Now, I am not going to test you on that concept. I just like to give a little extra detail about the cyclin-dependent kinases and the cyclins, please. Exactly. The cyclin-dependent kinase concentration is constant throughout. The cyclin concentration goes up and then down. So it gets degraded at the end. It gets degraded, I think that it's right after the assembly of the mitotic spindles. It starts to get degraded, if I'm not mistaken. I could be wrong on that. But somewhere in M phase, degradation begins. And then you have a low level in the new cells that result from the split. And then that starts to ramp up again at the end of the S phase, after the DNA has been replicated. Oh yeah, I think let me clarify it like this. When the cyclin and the cyclin-dependent kinase come together, they form something called the maturation promotion factor. It's really just an activated enzyme, right? It's an allosterically regulated enzyme that's now been turned on, okay? The maturation promotion factor level is what actually is going to trigger the cell division, all right? So if you have just cyclin-dependent kinase or just cyclin, the cell won't proceed through the M phase. But if you have maturation promotion factor, the combination of cyclin and cyclin-dependent kinase together, that is going to encourage movement from the G2, movement through the G2 checkpoint, and movement through the M phase.
So the purpose of all this is just to get the cycle.
The purpose is, if I can make all this cyclin, I can also be making everything else. So the cyclin could be considered as sort of an indirect measurement of all of the other activities inside of the cell.
So it just assists with the cell cycle?
Yeah, here's the key takeaways. Purge everything. Cyclin concentrations regulate the cell cycle. More cyclin, the cell cycle goes forward. If there's no cyclin, the cell cycle can't go forward. Cyclin works by activating cyclin-dependent kinases.
Okay.
Good?
So cyclin concentrations just regulate the cell cycle.
Yeah, exactly.
It works because it binds to the CDK.
Yep.
The CDK is constant.
Yep, you got it. The enzyme's always there. The cyclin's the regulatory molecule. It goes up and down in concentration. Oh, no problem. And the whole reason to have any of this is just to be like, hey, listen, there are intracellular things that are going on to make the cell cycle move forward. There are also extracellular signals like growth factors, okay? That's a positive regulator of the cell cycle. And then there are extracellular signals that are inhibitory, like density-dependent inhibition. If a cell is surrounded by cells and it detects that, it won't continue to divide. And then here's another positive regulator of the cell cycle, anchorage dependence. If the cell is not anchored someplace, if it's free floating, it usually won't divide. So there are cells that move through your body, through your bloodstream, through your lymphatic system, okay? But they don't usually divide until they're anchored somewhere and they're stuck in their location. And then they will stop dividing once they're surrounded by cells, all right? And these are things that malfunction in cancerous cells, where they're no longer inhibited by being surrounded by other cells so they can form a tumor, and they're no longer reliant on being anchored in a location so they can metastasize and break off and move through your body and continue to divide as they're just free floating in your lymphatic system or in your bloodstream. all right? So growth factors, all right? There's nothing too surprising here. Growth factors are chemicals that when you put them on a dang cell, it makes them divide. It's an extracellular signal that encourages the cell cycle, all right? Your body naturally produces hormones that act this way. Human growth hormone, right, works like this. It encourages cells to divide to continue dividing. You get more cells. Anchorage dependence. If the cell's not anchored, it won't divide. Density-dependent inhibition, okay, is beneficial because look at this sheet of cells right here. If some damage happens to a sheet of cells, you want density-dependent inhibition to be lifted so that the cells will then fill in that space. But if density-dependent inhibition doesn't work, the cells will just keep dividing and stacking on top of each other, making big blobs of cells. And then you end up with accumulation of waste products here, and then accumulation of waste products and a depletion of resources in cells that are in the center. And then they start putting out signals saying, hey, we need more resources. And then blood vessels can start to grow in here. It becomes a whole thing. All right. So anchorage dependence, it usually requires that a cell be anchored someplace before it'll start to divide. And then density-dependent inhibition, once the cell is surrounded by other cells, it will stop dividing. So you don't get the pile. Cool? And also, with density-dependent inhibition, if some cells get removed, I'm like, oh, there's open space there. I'll just divide and fill that space in. But I'll stop dividing once I'm surrounded by cells on all sides. That's the last slide of mitosis. Moving on, meiosis, all right? So I've said the photosynthesis respiration thing too many times. Meiosis is like 2 rounds of mitosis, basically, all right? Except in the first round of meiosis, you separate homologous chromosomes. And in the second round of meiosis, you separate sister chromatids. So in a perfect world, everything that I've said to you so far, every piece of terminology is just cemented in your brain, and that statement that I made complete sense. But we don't live in that perfect world, and that's okay. You know where we do live is in the best of all possible worlds, according to Camus, right? No, it wasn't Camus, Voltaire. Did I seem smart? The great thing about reading Voltaire stuff is it's super short, and then you can like quote 2 things from it, and people think you've read books. That's a pro strategy for all of you. Then you can go back to scrolling, which is way more rewarding in my opinion. All right, meiosis. It's like 2 rounds of mitosis, except in the first round, you separate homologous chromosomes. In the second round, you separate sister chromatids. So the second round is closer to mitosis there than the first round. Check it out. 2 rounds of division. Here's the first round, boom. Here's the second round, boom. Each one's got a prophase, a metaphase, an anaphase, a telophase, a cytokinesis. They're called like prophase 1, metaphase 1, and then prophase 2, metaphase 2, et cetera, et cetera. All right, so if you were cool with mitosis, then meiosis should be a little more approachable. All right, so why does meiosis exist? Mitosis can carry out reproduction, right? Bacteria don't carry out meiosis. They just do mitosis, and they divide, and they do just fine. All of your different cells were formed via mitosis. So why can't I just pop out another offspring? Why can't I just carry out asexual reproduction? Well, a long time ago, sexual reproduction began to exist, okay? Sexual reproduction evolved because it generates higher diversity. And more diverse populations are more likely to be able to survive changing conditions. If the population has a lot of variability, natural selection can occur. And if natural selection can occur, then a population or a species, whatever you want to say, population's better, a population can become better and better and better suited to its environment. All right. Now we're in an environment where things are changing and things are competing. So it's like you have to keep evolving just to stay in the same place. And that's the Red Queen hypothesis there, is that because of all the competition that's going on, it's like a biological arms race. That's terminology is thrown around sometimes. There are all these pathogens and competitors that are becoming better and better and more evolved to, say, eat us or to make us sick and kill us and eat all of our cells. All right? So we need to keep evolving. We need to keep generating variability so that our offspring can have some defenses against those threats. All right? So in order to stay in the same place, you have to keep changing. It's all about generating variability. This is where I'm going to recommend a book that's listed as a required reading in your course. Please, a question.
I was going to say, I learned there was a whole theory on why sexual reproduction evolved to be like one of the dominant forms of reproduction.
Yeah.
With parasites having trouble adapting to the variability of organisms.
Absolutely. Absolutely. So that's that biological arms race thing, right? About the, bless you, about the species being, you got pathogens trying to kill you, all right? If you and every other member of your species is genetically the same, if the pathogens can develop a way to kill one of you, then they're going to be able to kill any of you, all right? But variability means that some of you will have defenses. Yeah. So there's a lot of pressures. This book's listed as a required book for your course. It's one of my favorite books. I assign it. It's not one of my favorite books, but it's like a great teaching tool. I assign it every time I teach Bio 105. If you buy it, now's the time to get it, honestly, because we're going to start getting into genetics. And this book covers essentially the first half of a 300-level genetics course, but it's all in a graphic novel. So it's very digestible, all right? Like here's what some of the stuff looks like inside of it. It's great. Yeah, you can get a copy for $6. All right, and hey, here's the deal. At the end of the semester, if you don't want it anymore, I will give you $5 American for it. All right, I'm trying to build up enough of a collection that I can like 1 year donate or loan out all of them to students. So go buy the Stuff of Life. It's great. It saved me when I was taking my genetics course here in 2000 whatever, you know, and it covers like everything. And it's all framed by this alien asexual race that's dealing with the, you know, the bummers of being clonally reproduced. So it all relates to what we're talking about right now. The Stuff of Life, that's what it's called. A question.
Also, if you don't want to buy it on Amazon, Thrift Books has it.
Yeah, don't buy it on Amazon. I'll tell you that much. Like, stop using Amazon. I canceled my Prime. It was hard at first, but I get to feel better than so many other people, which is awesome. There's tons of other sources for the Stuff of Life, and I don't encourage you to purchase it on Amazon. I encourage you to purchase it someplace else.
Look at that, Thrift Books.
There it is, Thrift Books.
$4.69.
$4.69, 15 paperbacks, hardcover. The hardcover is like, you know, it's interesting. It's a good book. Check it out. Okay, cool. Let's go ahead and get back into meiosis. Yeah, thanks for bringing that up. Amazon's always the first result, but I'm willing to go out on a limb and say that if you can, you should try to move away from using them for things. But anyway, homologous chromosomes. We've already talked about them. Let's talk about them again, okay? Remember, you've got a chromosome from the sperm cell. You got a chromosome from the egg cell. When they fertilize, you end up with a copy of each chromosome, one from the sperm, one from the egg, now in the diploid organism, right? Okay, cool. So that means that each homologous chromosome pair, those chromosomes are referred to as a homologous pair, all right? 2 copies of chromosome 1, a homologous pair of chromosome 1. Each of those homologous chromosomes has has the same gene at the same location, usually. So like the gene for determining eye color. If they're different forms we call both of these alleles, they're different forms of the same gene. So we've got a brown eye allele and a blue eye allele. All right? Cool? I know I laid it all out for you before, but now it's going to become very relevant. All right, incredibly relevant. Okay. Oh yeah, and the location of the gene on a chromosome is called its locus. So the gene for eye color is at this locus on each homologous chromosome. All right. Sister chromatids, replicated chromosomes, it's all going to become important. Remember, a replicated chromosome and a chromosome are both called chromosomes. Non-replicated chromosome, replicated chromosome, it's still one chromosome. All right? This is going to get tricky. Bear with me. Here's a cell, all right? It is in G2 of interphase. It's preparing, it's getting ready to do its division thing, all right? Sister chromatids, non-sister chromatids, pairs of homologous chromosomes. I'm going to move past it unless we've got any questions. Okay, cool. Life cycle. Here you've got the diploid adults. One of these adults has ovaries. One of these adults has testis. All right? Meiosis is going to occur within the germ cells, within those organs. Okay? And after meiosis occurs, you're going to get sperm cells, the microgametes, and egg cells, the megagametes. Following meiosis, you have gone from a diploid organism with 2 copies of every chromosome to a haploid, say organism, but a haploid thing, all right, with 1 copy of each chromosome, all right? The egg and the sperm cell undergo fertilization, generating the diploid zygote, and the whole thing repeats. This life cycle that humans have, it has a diploid dominant stage where the haploid organism is not even really free living. Sperm cells don't live freely for very long, and egg cells, they never even really, they are fertilized internally and then undergo the first stages of development internally, all right? So the haploid stage of the life cycle exists, but it's incredibly reduced. And we see the same thing in most sexually reproducing plants, all right, where they've got these gametes that they generate, these haploid gametes, but the whole fertilization event and then the development of the diploid stage, the haploid stage is very short-lived, probably not free-living, and then the diploid stage, all right, it begins its development retained within one of the diploid adults. So there's this thing called the alternation of generations, where you have a haploid stage of the life cycle and a diploid stage of the life cycle. And some organisms have free-living haploid stages. Some organisms have only free-living diploid stages. It's a very interesting thing. So here are some examples of other life histories. Here's a mouse, all right? It does what we do, okay? Generates gametes, they fuse, fertilized internally, no free-living haploid stage. Now there's also things like the bryophytes, all right? If you've seen a moss out in the woods, sometimes it's just a soft green moss. Sometimes it's got these little stalked structures projecting off of it. You know what I'm referring to? You've seen them around here? Okay, so the majority of the tissue of a bryophyte is actually haploid tissue. So it's actually gametophyte tissue, okay? And only after fertilization do you get this very short-lived diploid stage, all right, which is the stalk. So that stalk and the little capsule on the end is a different, genetically distinct individual from the haploid stage, the gametophyte stage. Now, it's reliant on the gametophyte stage for resources because it's growing out of it, but it's the only diploid phase of that life cycle, so it's more of a haploid dominant life cycle. Yeah, please. No? Okay, cool. And then we've got other species that are way more haploid dominant, where they've got a very, very brief existence as a diploid zygote, which then immediately divides, all right? Sometimes the diploid zygote is even retained in like a colony of single-celled organisms. It's very fascinating stuff. You'll talk about this a lot if you take Botany 105. Here, I just wanted to I like to occasionally because your textbook's written like humans are the only things that exist. And I just like to point out, like, okay, there are other life cycles, but they all the sexually reproducing organisms undergo some alternation of generations. All right, where they have a haploid stage and then a diploid stage. That's what allows for the generation of variability. Okay, meiosis generates gametes that are distinct from the parent, and then those two gametes fuse to make an individual that is distinct from either parent. It's the whole purpose, is to make distinct individuals and generate variability. Okay, so all this stuff has been why have sexual reproduction? It's all about generating variability. Then I give you an opportunity really quickly to touch base on homologous chromosomes, sister chromatids, non-sister chromatids, diploid, haploid. I just want to touch base on all that stuff. Yeah?
So, in humans, there's 23 pairs of chromosomes.
Yeah.
So chromosome 1, there's 2 chromosome 1s. Is that they're both homologous?
Yes, they are homologous to one another.
And that's because I got 1 chromosome 1 from my dad, 1 chromosome 1 from mama.
You got it. And they should have the same instructions on them. They're probably going to be like 99% identical to one another. There can be some variability, all right, where you have a different allele. Like I have the brown eye allele. My mom gave me the brown eye allele. My dad gave me the blue eye allele. And now I have both alleles. And which one's going to be expressed? And that has to do with our later discussions of dominant and recessive characteristics and polygenic inheritance and all those things. But yeah, you've got it. The homologous pair is because you got one from the sperm cell, one from the egg cell. And of course, as we move forward with technologies, there are going to be variabilities to this, But the key is that you go from a diploid cell to a haploid cell, all right? And then 2 haploid cells are fused and generate another diploid cell. All right? Awesome, please.
Can you go back to the other slide? I feel like
This?
no.
This.
Yeah. So is there like a relationship between homologous chromosomes and non-sister chromatids? Are they the same thing?
They are not the same thing, but once okay, here's the real stinker, right? Once these get pulled off,
Yeah.
they're then going to be homologous chromosomes. Right now, this is a homologous chromosome, this is a homologous chromosome. The sister chromatid terminology is specific to when the chromosomes are in their replicated stage. All right?
Okay, so when they're replicated, they have sister chromatids.
Exactly. Sister chromatids.
Chromatids, sorry.
No problem.
There's too many words.
I know. I just want to make sure that our terminology is very, yeah.
And then when they pull off sister chromatids, would those 2 non-sister chromatids going to be a pair of homologous?
They're not going to join, but they are homologous. So check this out, right? It's bad. It's not great. Just bear with me here. Oh, wait. I remember I can use I'll use the document camera. It works. That way I can, do it right here. Okay, document camera. And this stuff is super freaking tricky. So no worries. It's good that we're going over this. Now, does this have anything in it? What is this? Oh, sick. Now, does that work? It does. Oh my gosh. Everything's coming together. Perfect. Okay, great. So, boom, boom. Is that legible? Can everybody see that?
Can you turn on the light a little?
Ugh. You can't see the color or anything. That's working better. That's starting to work. Oh, that did it. Yeah, there we go. Crazy. All right. This right here is a homologous pair of replicated chromosomes. All right, this is the story of a homologous pair of replicated chromosomes. All right? Boom, 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 boom. Sister chromatid. Boom, boom. Sister chromatid. Non-sister chromatid. Right? So far, so good. Now, we're going to divide these. I'm going to put a little division symbol here and a little division symbol here. Boom, boom. And I know you can't see the bottom part, but okay. So we've got this, and then they're going to divide. They're going to divide, and they go, there we go. They divided, all right? Now, I'm going to say right here, boosh, 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 boosh. Homologous chromosomes. As long as you're comparing one of these black chromosomes to a red chromosome, that's homologous pair, that's a homologous pair there. These are homologous pairs, these are homologous pairs, that's a homologous pair, and that's a homologous pair. Does that sort of make sense? But now there's no sister chromatids here at all because everything's separated.
So the black and the red one together.
A pair of homologous chromosomes. But they're not going to join together or anything. They're just going to be, they're homologous pair. They're homologous to one another.
Okay, the black and the red one are homologous to one another, but they're not going to join.
No. Because later what's going to happen is you're going to actually divide these up into 4 different cells.
Oh, and then that.
That's going to fuse with other gametes. These are now the gametes.
They're just homologous because they're like. Trying to think of the right definition.
I'm gonna try to cut you off. They're all at chromosome 1. They all have the same information on them. They're all, yeah.
Those two pairs were both chromosome 1.
Yeah, chromosome 1 and chromosome 1.
So they're homologous to one another because they're two pairs from different, let's say, a mom and a dad.
Yep. So this came from the sperm. This came from the sperm cell. This came from the egg cell. And then during S phase, they replicated.
Okay. But they're from two different people, so then they're homologous.
They're homologous, exactly.
Okay.
Because there's going to be some differences, but they're still very similar to one another.
They're the same type of chromosome pair, just from different people. Okay, I got it.
Awesome.
Great, thank you so much.
No problem. We got there. Whoa, crazy.
It was crazy, thank you.
No worries. That was fun. Okay. So let's go ahead and just get into the stages of meiosis. All right. It's got a prophase, a metaphase, an anaphase, and a telophase. Why is there a play button on this? There's no reason for this to be a video. Interesting. Well, either way, there are stages of meiosis. You've got prophase, metaphase, anaphase, telophase 1, prophase, metaphase, anaphase, telophase 2. All right? So you have meiosis 1 and then meiosis 2. Meiosis 1 and meiosis 2 each have prophase, metaphase, anaphase, telophase, cytokinesis. All right? So each one has the full mitosis, all those phases of the M phase. Cool? Awesome. So in meiosis 1, the homologous pairs come together and then they separate. Everything's falling apart. Meiosis 2, there we go. So in meiosis 1, everything is that legible? Cool. In meiosis 1, everything's in the replicated stage. Okay? and they are going to separate. So you're going to end up with one cell that's got the replicated chromosome here, and one cell that's got the replicated chromosome here. In meiosis 2, you're going to take that situation. This is one cell. Okay. You're going to take that situation and then separate the sister chromatids up. Okay, so in meiosis 1, which is also called reductional division, you've got a diploid cell, which then divides, and the homologous chromosomes get separated. So therefore, it's now a haploid cell. There's only one copy of chromosome 1 in each of these cells. And the same thing happens for all of the other chromosome pairs. Okay, so meiosis 1, you've got homologous pairs of chromosomes get separated. And then in meiosis 2, all right, the sister chromatids get separated. When a cell is entering meiosis 2, it has no homologous chromosomes. All right? It has one copy of every chromosome, assuming that meiosis 1 went well. All right. Now, there's some additional pieces here. For example, in meiosis 1, there's actually some exchange of information between the chromosomes. So you can end up with swapped out regions of the chromosomes like this. They call them recombinant chromosomes. You can see that some of the material from this red chromosome moved over to the black chromosome and vice versa. So you're going to end up then with, additional complications that carry through, or additional things that carry through the entire process, like this, where now the chromosomes are no longer identical to how they were in the sperm cell and the egg cell that generated the individual, you see? But that'll come up very soon. Yes?
So by this process, that's what produces variation in individual gametes?
This is one of the that produces variation in the individual gametes, yes. But also, think about this. For each pair of homologous chromosomes, right? You could have the maternal chromosome 1, paternal chromosome 2, paternal chromosome 3, maternal chromosome 4, maternal chromosome 6, paternal chromosome 5. So you're going to have in the gamete some purely maternal DNA and some purely paternal DNA on a chromosome by chromosome basis. You see, so that also generates variability. In my cells, I've got both the sperm DNA and the egg DNA inside each cell, and it's all being read and expressed. But when I generate a sperm cell, it's going to have some of the sperm DNA in me and some of the egg DNA in me, but it's not diploid. So it doesn't have all of the DNA that I had for chromosome 2 in it. It only has either the sperm or the egg DNA that I originally had in me, you see? So that's what really is going to generate the variability. All right. I mean, this crossing over process is really important as well. But the fact that you only get one of each of the chromosomes in each homologous pair is what really makes the gametes different from the organism producing them. Which, on that confusing note, that is the end of our time today. So we're going to wrap up, and we'll come back to all of this on Wednesday. I'm very excited. Don't worry, there's only 18 slides on meiosis, and we're just going to really take our time on it, okay? Then we'll get into some genetics. People seem to think that's pretty fun. I sure do. So, yeah, we got good stuff ahead. Keep up the great work, everybody. Thank you for making it to class today, and I'll see you on Wednesday. Goodbye. Have a good one.